important for rice production. Nitrogen is generally applied as ammonium sulfate or urea. The elemental nitrogen is also available in large quantity in the atmosphere, but the elemental nitrogen which is present in the atmosphere is not utilized by any cropping system. There are quite a number of microorganisms which can fix atmospheric nitrogen and transfer to crop plants. Particularly in rice field ecological condition, there are a number of nitrogen fixing organisms are available. The nitrogen fixing organisms like the free living cyanobacteria, the symbiotic cyanobacteria in association with Azolla, and quite few bacterial groups are also fix nitrogen here. So among the nitrogen fixing organisms in the rice ecological condition, the symbiotic system, the Anabina azolla, is a very effective one in rice field. Azolla is actually a water fern which floats in water and fixes the nitrogen. In this particular plant, the symbiotic nitrogen fixing cyanobacterium, Anabina azollae, is present in the leaf. The plant actually contains, there are two leaflets, one is the upper lobe and another one is lower lobe. The nitrogen fixation process is going on in the upper lobe, where the symbiotic cyanobacterium resides inside a cavity. Because of the nitrogen fixing potential of this particular plant, the total biomass production over a period of time is very high in rice soil ecology. Therefore, based on the growth rate, based on the doubling time, based on the nitrogen fixation potential, this plant may be exploited very well as a biofertilizer for rice production. There are quite a number of species are also available in Azolla. The one species, Azolla pinnata, is very popular in most of the tropical countries. At the same time, the Azolla folliculoides, Azolla microphylla and Azolla carolinea were very popular and occur widely in temperate countries. So for higher biomass productivity as well as higher nitrogen fixing potential, we have to select the best species depending upon the local situation. So once they fix nitrogen, the nitrogen is accumulated in the plant, the total biomass is very high in the rice field, and once the plant undergoes degradation in rice soil, then the nitrogen is released into the field. That means the nitrogen is released after mineralization in rice field, and also the rice plant can take very well the released ammonia for their utilization and the crop productivity. So in general, the Azolla, when it is used as a biofertilizer, this can contribute around 40 to 60 kg nitrogen. And when we looking onto the use of Azolla directly by the farmers, this can be used as a dual crop. One can grow rice as well as Azolla simultaneously. After rice transplanting is over, that is about 10 days or a week after transplanting, Azolla at the rate of 500 kg fresh biomass may be inoculated in the rice field. So the inoculated Azolla can multiply and develop in the rice field along with the rice for a period of 25 days. Then they can become form a mat in the rice field. Then after 25 days, the developed mat of Azolla may be incorporated in the rice field. That will supply the nitrogen, organic nitrogen, to the crop. The Azolla is inoculated in the rice field as a dual culture. Normally, Azolla can be used directly for incorporation before planting, as well as for growing Azolla along with the rice. The We have Azolla folliculoides as well as Azolla microphylla, the fast-growing types. The Azolla microphylla is a fast-growing one, as well as tolerant to high temperature. And the nitrogen fixation is also high in this plant. Azolla can be grown in field condition in larger plots and multiplied, and the biomass may be collected and used in rice field. For large scale multiplication, we can prepare 1 cent plots, that's 40 meters square. The field can be plowed and leveled very well, and finally the plots may be prepared and require nutrients to be applied for growth and multiplication. It's very important to prepare the field in a well leveled condition and then Azolla to be grown. Once the plot is ready for multiplication, the nutrient required for the growth are the fresh cattle dung 10 kg to be mixed in 20 to 25 liters of water, and simply broadcasting the fresh cattle dung solution. So this fresh cattle dung actually activate the growth and multiplication of Azolla. In addition to fresh cattle dung, Azolla also requires a good quantity of phosphorus fertilizer. For 1 cent plot, we have to give 300 grams of superphosphate for growth and multiplication. Even the 300 grams may be divided as one basal application as well as top dressing 2 doses at the rate of 100 grams. So in addition to the nutrients, the fresh cattle dung as well as the phosphate sources, there are few pest population also occur and damage the Azolla population. Therefore, during multiplication we have to add for 1 cent plot at the rate of 100 gram of furan granules to check the pest population. So after completing the nutrition as well as the nutrient application as well as the pesticide application, the Azolla, fresh Azolla to be inoculated into the plots. For 1 cent plot, 8 kg of fresh Azolla inoculum to be broadcasted. So during inoculation, care should be taken to spread the Azolla inoculum in a uniform way so that the multiplication will be fast as well as uniform. Once the Azolla is inoculated into the plants, then they try to multiply in the plot and just to float and accumulate as a mat of biomass. So after 15 to 20 days, depending upon the climatic condition and the season, normally this is adapted in winter season, the growth will complete in about 10 to 15 days or at the maximum 20 days. Once the mat is developed, then the Azolla biomass to be harvested from the multiplication plot. One can use very simply the 2 bamboo sticks and collect the material in plastic buckets or any other container, and the collected biomass may be transported to the main field for inoculation. Again here the biomass may be transported mostly in gunny bags, which is really suitable for transport purpose, not only for inoculation in the same area, but also when once farmers likes to transport this in a different area, they have to take it in gunny bags, which is a very suitable container for transport. Then once it is produced, then it should be inoculated in the main field. The main field after transplantation, that is a week or 10 days after planting, the Azolla to be broadcasted at the rate of 500 kg per hectare. plant. So initially the inoculated Azolla multiply and develop again as a mat in the main field. And at this point of time, the total biomass production per hectare is around 15 to 20 tons per hectare. So this is after inoculation and the full mat development in an inoculated rice field. Once the mat development is seen in the rice field, then it should be incorporated in the rice field, because the nitrogen accumulated in the plant as such is not made available to rice plant. So the Azolla plant or Azolla biomass to be incorporated in rice by manual labor, or sometimes one can use some intercultivator to tamble the Azolla. So during incorporation, also the laborers can remove the weed and the cost of cultivation may be considerably reduced. So both operation of weeding as well as incorporation may be contemplated to reduce the cost of incorporation. So once it is incorporated, then the entire biomass undergo degradation and mineralization, then it is made available to rice plant. And also for Azolla multiplication and development process, we need to select the best and efficient nitrogen fixing cultures. Therefore, we have developed a system called the culture collection center, and from there we can select a suitable culture and transfer to the field. In the Azolla also few pest population occur, particularly the pest called pyrolis also affect, and also snails can affect. The snails can eat and reduce the biomass. So this is a situation where a pyrolis pest, the larvae can affect and damage the plant considerably. Rather, it can decompose the plant very quickly once the pest population occurs. It's very interesting to note in this particular aquatic plant that they do produce the sporocarps. That the spores are produced, both the male and female spores, which are very minute structures, something like a roggy seed. The spores are there. So the spores, what we do, we just collect the spores in the laboratory condition and examine the spore under microscope, and also we are investigating carefully the type of spores, and also decay the spores along with the Azolla plant material under laboratory condition. Then once the decomposition is completed along with the spores, then we separate the spores and dry it in shade, and it is used for multiplication also. So it is very important to use the spores under natural condition, because the spores can survive in the field for a longer period, may be exploited very well as an inoculum. In addition to biofertilizer value, Azolla can also be used as a poultry feed, because it contains a very high amount of nitrogen and proteins in this plant. Apart from that, this can also be used as a cattle feed. Either can be fed with pigs or other cattle. This can be used. So generally in many other places they use as a cattle feed for higher production of pigs as well as other animals. So in short, when we use Azolla as a biofertilizer, we can get around 300 to 450 kilogram of total increased paddy yield when compared to the untreated Azolla plots. So the net income may be ranging from 750 to 125, and the total cost of production involved is only around 120 rupees. Therefore, the net income may be around 630 to 1000 rupees a farmer can easily attain by using Azolla as a biofertilizer.